The title is different, slightly different, because I promised to the people of satellite cells to talk about the displacement of myonuclei and myofiber regeneration in long-term human mass denervation that could be treated or not by electrical stimulation. And since I would like to save time, as I promised, I asked to all people, I present these summary slides, what is the behavior of a long-term denervated muscle. This is a control muscle. After almost 1 year of permanent denervation in humans, not in mice or rats, muscle fibers are still there. They are just atrophic, but they may respond to electrical stimulation, of course. Only 3 or 9, 10 years after denervation, the muscle seems to disappear. But this is a 3-year denervated human muscle taken from biopsy from those patients, spinal cord injury patients. It seems that there are no muscle, maybe a few big fibers. But what is the surprise? That if you go to a larger magnification, what appears to be connective tissue is a big number of very, very, very severe atrophic muscle fibers, from which all the contractile protein disappeared, and in which the nuclei grouped in the center of these very, very small tubes. These are not myotubes. These are severely atrophic muscle fibers that survive denervation much longer than what people usually think about. The same picture, yes. Notice that there are fibers with contain almost only nuclei, and you see here in a longitudinal section that these groups of nuclei are separated by stretches of without contractile proteins. This is a big fibers remaining. You see also in this myotube-like structure, there are nuclei grouped, and sometimes there are only nuclei. Here, here, here. This occurs typically from 3 to 7 years post-spinal cord injury. After 10 years, this phenomenon disappear. This is correlated to the fact that if you start at before 1 year the electrical stimulation, it is very easy to have response because you are dealing with just atrophic muscle fibers. If you wait 3 years, 5 years, you have to wait that these fibers recover their the synthesis of contractile proteins, and luckily they do. Here another example of a multinucleated contractile muscle fibers, very severely atrophic, and of course with a lot of dystrophic images. A second not expected phenomenon is that a few muscle fibers regenerate in these muscles. We believe that these green fibers of different size are regenerated fibers because they contain embryonic myosin. The green label is the stain due to immunofluorescence with an antibody against embryonic myosin. Since we know that spontaneously, even without the innervation, the muscle fiber, the new muscle fiber make a transition from embryonic myosin to fast-like adult myosin heavy chains, we may say that these fibers was born not earlier than 2 weeks before the biopsy was taken. This is a continuous, unfortunately not compensatory event, but if you go from a severe atrophic muscle, even starting from this stage, and perform electrical stimulation, long-term electrical stimulation, you may reach this situation. This is before electrical stimulation, a patient which was thigh was analyzed by CT scan and colored according to density of the muscle tissue. During the first year, the muscle is still there, but after 2 years, 4 years, 6 years, the muscle seems to disappear. What happens if you start stimulation during the first year? That 2 years after, and therefore at 3 years of denervation, you have an almost normal muscle. This is true also if you start from 2 years. It is astonishing, but true that this occurs even if you start later, 4 years. Of course, if you wait more than 5 years, the results are still impressive, but not the same. We don't believe that those are reinnervated fibers, because if you discontinue electrical stimulation, this patient have to do 5 times per week, 2 times a day electrical stimulation to mimic what is the trophic effect of the normal innervation. Those effects disappear. More, they need a kind of the amount of current to stimulate the whole muscle in the thigh, which is 100 or 1,000 times more than the current needed to stimulate through the nerves. Therefore, since they never achieve this sensitivity to electrical stimulation, we are sure that these are denervated muscle. We have very good correlation between CT scan area and denervation time before, after electrical stimulation. We have a good correlation between denervation time and fiber diameters before stimulation, after 2 years of stimulation. There is a good correlation between the zero strength of the denervated muscle and the force that is provided by this muscle after 2 years of denervation. The force is enough to let this person, you see here the patient, is able to stand even without the 2 hands, because those muscles are functional. It is not big tissue, but it is big tissue, that muscle tissue that may provide external work. I am earlier than 20 minutes, I hope, and therefore
Sure.
I thanks the audience for attention.
Sure you are. Thank you. This was the fastest talk today. Thank you, Ugo. I guess there are some questions to this interesting talk. Don't see any.
Ugo, it's strange that I ask a question because we have done a lot of this work collaborating, but can you tell me something more or tell the audience something more about this positioning of the myonuclei? Do you think
first, let me say, I demonstrate what is a denervated muscle after years of denervation, and what is a denervated muscle still denervated but stimulated by during to the additional year, and these are of course Feliciano's and Simona Boncompagni electron microscopy, and they have to be normal, because otherwise the muscle do not contract. Feliciano, question, please.
Tell me something more about what you were saying about the positioning of the myonuclei, because that's something that I never studied, I never paid attention to it, but we know that myotubes have central nuclei. Then when the fibers grow, the nuclei are pushed toward the periphery. And I don't know much about this mechanism, you know?
Yeah, but no one knows exactly, even Ugo, but this could be previously a myotube that is developing in a big fibers. But this is not the usual position of the nuclei in the myotubes. In the myotube, in the real myotubes, they are one near the other continuously. You agree?
Yes, yes, absolutely.
Yes. These are old muscle fibers that are atrophic at the point that they have no more contractile protein inside. For an unknown reason, the nuclei tend to groups. Why? I don't know. But these are evidence that the fiber are denervated since 3 years, 4 years, 5 years.
There is a very talented scientist from Scandinavia. Now I don't remember the name. I think from Sweden, which has done a lot of these studies about the memory of muscle. Yeah. So the muscle have memory of exercise, and the memory of exercise is in the nuclei, and the nuclei survive with this memory for long time. And
I fully agree.
And this is, I mean, your fibers, the denervated fibers regenerate under functional electrical stimulation because of those nuclei being alive, you know, and I think it's a very important point.
Yes. We have an experiment in rodents, in rats, long-term denervation. In rats means up to 1 year, 2 years, not more, because they do not live more. And always there are fibers that are unable to contract, but if you impel them by an electrode, they have all the performance of sarcolemma and control of calcium inside, even small like this.
Well, if you remember the work we had done with Fabio Francini in Florence,
yes,
I remember still
we have evidences that the denervated muscle fibers survive much longer than what the people believe.
Yeah, they lose contractile elements, but the electrical properties stays there for a long time.
Yes.
As the nuclei, even longer maybe.
That's all.
Yeah. Yeah, this is Terry Partridge. The work, I think it's Gunderson, the man in Sweden who looks at myonuclear positioning and number, and he does this in the mouse. And I think mouse and man are quite different in this respect. So in mouse, virtually all regenerated fibers develop a strong central nucleation, and they don't lose it for most of the animal's life. So it is a very persistent thing in mice, and it doesn't seem to be so persistent in man. I also I think that, I'll talk about it a bit tomorrow, that the mouse routinely, when it regenerates, doubles the number of nuclei per fiber, and a lot of those are central. And that, again, we don't know whether that happens in any other species.